All right. So let's talk about amides or amides, however you want to say that. I say amides because it's easier to hear the difference between amine and amide, amine and amides. So Tylenol, an aspirin substitute, contains acetaminophen. Acetaminophen is an amide. And the functional group for amides, again, are C double bond O attached to a nitrogen. Now, acetaminophen, we get to make it. Well, we don't get to make it. We would have made it in lab. I think you'll probably watch a video on it. It's a fever reducer, pain reliever. Um, Actually, this is Tylenol, acetaminophen. So I think I'm thinking of acetylsalicylic acid, which is ibuprofen. That one has anti-inflammatory effects. Tylenol doesn't have very much anti-inflammatory effects, but acetaminophen nonetheless is an amine, and you can also see why it might be bad for your stomach. It has a phenol group on here, which means it's acidic, and so this can often they maybe buffer these with some kind of base, but this could upset your stomach, give you some ulcers or something. So some preparation of amides. How do we make amides? Well, they're made from carboxylic acids that get reacted with nitrogen containing compounds or amines. And we can react them with primary or secondary amines. Now, what will happen is that um, the nitrogen will replace the OH and will just connect whatever was attached to. Well, actually, I guess this isn't showing a reaction. This is showing what changes. It's saying the OH changes to this. Let's look at a reaction so you can kind of see how it happens. So what we're going to keep from our carboxylic acid, of course, is everything without the OH on it. And then we're just going to remove one of the H's from our primary or secondary amine and add that carbon to the nitrogen. Now, this works with primary and secondary amines, but not with tertiary amines, because tertiary amines don't have any hydrogens to remove. So methylamine isn't exactly a great example, but even if I showed you dimethylamine, you can see that it has an H. And so dimethylamine would just turn into this guy, as opposed to having just the H on it. But we would still form the bond between the carbonyl and the nitrogen, and we would have our amide. Naming, we change the last, so propane gets the E dropped, and we make it propanamide, just kind of like propanamine. Now, these reactions are called amidation. We also call it condensation reactions, because I guess, because they produce water, maybe, or they condense into one molecule. I'm not really sure on the origin of that name, but amidation makes more sense to me. And these work best with carboxylic acids, primary and secondary amines, and a little bit of heat. Can add some energy. Now, we don't go into all the mechanisms here. There are some. It is a little more complicated than that, but we won't go into all of that. Now, yeah, let's do an example. So let's say you've got these two, carboxylic acid and amine. Predict the product. So your job here is to draw what we're going to get. So remember, you are removing the OH, you're attaching the N, and we're losing a hydrogen. So draw it. It might be easier for you guys to set it up with a condensed structure when you're drawing them. That way you can lose your water and then just see how everything else should connect. Um, so hopefully you did something like that. Now, let me think. Again, with substituents, this doesn't get harder. You just have to keep track of where you're making your attachments. So if we had, you know, something like this, all of this is just going to be attached to the N. We just need to make sure we're not losing anything when you sometimes students will attach this to that, and then over here the structure looks different because maybe they weren't paying attention to, they're so focused on making the right connection that they don't draw the molecule the way it used to look. I'm a stickler for that kind of stuff. Your molecule shouldn't change. If it does, that makes me think maybe you don't know what you're doing, or yeah, that you made a mistake because you weren't paying attention. When we name amides, we can, in both the common and IUPAC names, amides get named by dropping the oic acid or the ic acid from the carboxylic name and adding the suffix amide, basically. So this is butanoic acid, so then it would just be butanamide. Or butyric acid, common name, butyramide. So again, it's kind of like taking the parent name and dropping the E like we did for amines, but here, because the parent name ends in oic acid or ic acid, we're dropping that part of it. So let's see. All of these are examples. So let's. This was formic acid, common, or methanoic acid. So now it's methanamide. This was ethanoic acid, ethanamide. This was benzenoic acid, benzamide. Again, C double bond O and the amide. There could still be substituents on the nitrogen. So if that happens, we have to go back to the NN business. So here we have NN dimethyl. So this is just NN dimethyl butanamide. So same parent name as before, butanoic acid or butyric acid, and then say the NN first, just like we did with our amines. So very, very similar naming convention here. Shouldn't be too much to have to figure out. Quick learning check, or I guess a work through, but you can treat it like a learning check. Name this one. 4 carbons, it's got a substituent on the N. So don't forget to say N-methyl, and then butanamide or butyramide, if you were using butyric acid, common name. A couple more. Give the IUPAC and common names for the following amides. I'm less worried about the common names because I don't think I really emphasize the common names of the carboxylic acids. But pause it, give it a shot. So ethanoic acid becomes ethanamide or acetic acid. And then and this one was a little more challenging, right? N-ethyl propanamide, propanoic acid. Good. Physical properties of amines. Amides do not have the same properties of bases. So they are not bases. There's no lone pair, so they don't do that. Only methanamide is liquid at room temperature. All other amides are solid. Primary amides have the highest melting points. And that's because the NH2 group can form the most hydrogen bonds. The secondary amides are slightly lower because they only have, there's some other group on there, only one H. And then the tertiaries, 2 groups. I don't know what my R's just mean groups, I guess. 2 different ones, R1, R2. No H's, so they can't form any hydrogen bonds, and so they have the lowest of the melting points. As you can see that here, a nice comparison. 80 degrees, 28 degrees, and then negative 61. Like, again, a huge jump with H bonds in terms of melting temp. All right. and this actually shows it a little bit better. So here you can see with water, so this is solubility. Primary amines can form hydrogen bonds, not just with water, but with other NH2 groups of other amines and other amides as well. So they would be soluble with amines and in water, so long as they have 5 or less carbons. They should be pretty soluble. Again, nitrogen can make a bond, and then the two hydrogens can make more hydrogen bonds. Secondary, this kind of slows down a little bit. So we can do one with a hydrogen and one from the nitrogen, but that's about it. And again, they're soluble in water and other amides and amines because they have limited hydrogen bonding. And tertiary, these tertiary ones, lowest melting points because they don't have that hydrogen bond that they can form with each other, but they can still form hydrogen bonds with water, only one. They're not as soluble. Um, so like if you had a 4 carbon amide, a tertiary amide, that one might not be as soluble as a 4 carbon primary amide. You know what I mean? So that can hurt it a little bit. But for the most part, we'll say 5 carbons or less, you're soluble if you are an amide. Urea, the simplest natural amide. This is the end product of the protein metabolism. Um, this is what your body excretes, and basically, it's an amide. You can see here it's actually like a diamide. It's a amide functional group in this way. Two molecules of ammonia basically reacted on this acetic acid, or sorry, on this formic acid. And yeah, there's a condition called uremia if if this actually builds to a toxic level or to high levels. So um, just relating some amides to your body. Aspirin is another amide. They can contain different kinds of compounds, aspirins. Phenacetin and acetaminophen are both examples of the kind of compounds that aspirin substitutes. These are aspirin substitutes. Aspirin, I think, is acetylsalicylic acid. Unless I always confuse that with ibuprofen. Sorry, I'm just kind of thinking out loud. Both of these have fever and pain reducers, not so much anti-inflammatory effect, but again, they're amides. That's what we're looking at here. Phenacetin and acetaminophen. Barbiturates. These are cyclic amides of barbituric acid. I don't think I have that acid shown. But they're just amides of that acid. So that acid reacted with different amines. So you can kind of see those here. Let's see, where can we find? Actually, it was hard to see where the amine group is getting added on here. No worries, we don't have to make sense of that, of these structures. These barbiturates can be sedatives or sleep inducers in large doses. They are habit-forming, phenobarbital, pentobarbital. Again, amides make good medicines. Let's talk about hydrolysis of amides. So in a reverse reaction of amidation, we can hydrolyze these. So we can put water back and actually split them back into our acid and our base, or what we can say, we get back our not our acid and our base. Well, yeah, I guess we do. We get back our carboxylic acid, and we get back our amine, which is a base. But it's going to depend on the on the pH, whether or not we get certain salts. So we're going to take a look at that. So depending on what we react this with, we can split we can actually hydrolyze acids. Wow, man, I'm just like so jumbled with the 3 slides that I'm trying to get to right now. Let me just take a second to back up a second. We're going to write reactions for hydrolysis. We can hydrolyze using acid or base. Let's not confuse that with the fact that the hydrolysis products are going to be acids and bases. Okay, the acid and base that we hydrolyze with is separate from the fact that we're dealing with acids and bases here as products. Carboxylic acid, base in the amine. Okay, let's start with acid. So let's look at acid hydrolysis. So again, hydrolysis products for amides are always going to be the carboxylic acid and the amine. But depending on pH, we need to see what's going to happen to both of these things. So in acid environment, we're going to split our amide right down that center bond. We're going to put back an OH on our acid. Now, why are we going to put back an OH on our acid? Well, you remember when we did saponification, when we were hydrolyzing our fatty acids in the carboxylic acid chapter, if we did it with base, when we put back this OH, the base would then strip an H off of it and then leave it with an O minus. So we always put back the OH to get back our starting carboxylic acid, but then we go and say, okay, what is the environment going to do to this OH? In an acid environment, that's always just letting everybody grab H pluses if they want them, this OH is going to look just like that. So we'll have our actual carboxylic acid back. In the amine version, because the amine is going to form like this with a lone pair, we have to ask what's going to happen to that lone pair in the presence of acid, where everybody who wants an H can get an H. It's going to add an H. And so that's why we see it with an extra bond to an H, a positive charge and a counter ion. So in acid, you can memorize this. We're going to get back our carboxylic acid and we're going to get an ammonium salt. This will be different than what we get in base. Let's take a look at the base situation. In base, we are still going to cut our amide right down the center. We're still going to add an OH to the side of the carboxylic acid. And then we're going to think what's going to happen to it. Well, in the presence of base, where this is always trying to strip H's off of anything acidic, our OH is going to lose its H. We're going to be left with an O minus. The counter ion will pair with it. So we'll have a carboxylate salt. Acids don't live in base, just like bases can't live in acid. That's why back here, this base couldn't live in acid, so it became an ammonium salt. This acid can't live in base, so it became a carboxylate salt. The amine, on the other hand, just like the acid in the previous example, is right at home in this environment. This amine is basic. This is a basic environment. Nothing is providing hydrogens. They're taking them away. That's what bases do. They take hydrogens away. So the methylamine is just fine. So while you always will get some version of a carboxylic acid and always some version of ammonia, whether that's ammonia or ammonium, I guess what I'm really getting at here is an amine. You'll always get some version of amine and some version of a carboxylic acid. So you just have to pay attention to which one you're actually dealing with. So quick learning check. Draw the condensed structural formula. Write the IUPAC name for the products for the hydrolysis of N-methylpentanamide with sodium hydroxide. I'll help you get started. 2, 3, 4, 5. Pentanamide, N-methyl with sodium hydroxide. So it's in base. First, just figure out in base, what are you going to get? You're going to get a carboxylic acid, or you're going to get a carboxylate? Are you going to get amine, or are you going to get an ammonium salt? And once you've figured out which one you're expecting, cut your molecule in half, add the parts back that you're supposed to add. All right, give it a shot. So hopefully you remembered that in base, acids can't live. So we would have gotten this sodium pentanoate. Our pentanoic acid would become sodium pentanoate. And our methanamine stays methanamine. We don't have to call it N-methylmethanamine. It's just methanamine. Let's try another one. This time an acid hydrolysis. So acid hydrolyze this amine. And you can name them too. Why not? All right, give it a shot. So here cutting this in half, remembering that in acid, we're going to get our carboxylic acid back, so propanoic acid. And then in acid, our amine, in this case methylamine, is going to become methylammonium. And not just any methylammonium, methylammonium chloride, because that was the counter ion for our acid. All right, good job. Now, for the remainder of these slides, I decided to give you guys a lot more info than I normally would, because there's a lot of info here, and I didn't want to have to write it all down. So we're going to talk about neurotransmitters. Neurotransmitters are chemical compounds that transmit impulses from a nerve cell to a target cell. And so the way that they work is the nerve impulse, we're not going into that really, is an action potential that travels along the membrane here. When it reaches the end, it triggers the release of these vesicles. And these vesicles then move towards what we call the synapse, which is the gap in between nerve cells. So this is the one end of the next nerve cell, and this is the end of the one sending the signal. And they dump all of these neurotransmitters, little chemicals, into the synapse, into the gap. And those synapse gaps are lined with receptors. And when those chemicals match up with their receptors, they now start a new signal cascade in the next neuron. So this is how cells talk to each other. And of course, these can be very branched. They can talk to lots of cells at once. These are all insulated by those myelin sheaths. But again, I guess this picture is also good. So here's the body of a nerve cell. Let's say signals are coming in from somewhere, triggers a signal down the length of this nerve cell to then talk to neighboring nerve cells, and then the whole process starts over again. Now, neurons are what these nerve cells are also called, have, again, a cell body, numerous filaments called dendrites. They have the axon at one end and axon terminals at the other end, and the junctions in between called the synapse. Now, the neurotransmitters at the synapse get released directly into the synapse and can be taken up by the neighboring nerve cells, and then they get moved through those nerve cells. Now, electrical impulses and chemical transmitters, they move very, very quickly. They go through this network of cells in a very short amount of time. The neurotransmitters themselves can be either excitatory or inhibitory. Excitatory means that they stimulate whoever they talk to send more impulses, more impulses, more impulses. So tell everybody. Inhibitory, whenever a nerve cell receives an inhibitory neurotransmitter, it sort of shuts it off, makes it quiet, so that it doesn't keep exciting other nerve cells, and it kind of kills the signal. And we're going to look at examples of both types of neurotransmitter. The binding of an excitatory neurotransmitter can open a channel in the nearby nerve cells that release more neurotransmitters. So in that way, the excitatory ones are really good at sending a cascading signal, kind of a chain reaction. So neurotransmitters can be removed a few different ways. One, once they get dumped into the synapse, they just slowly kind of spread out until they're no longer in the synapse. And then when they're gone, the signal stops. They can be broken down by enzymes. So sometimes as the neurotransmitter gets dumped into the synapse, an enzyme is actively going and then breaking them down so that the signal again stops. Or they get reuptake. What that means is that some mechanism collects them and then puts them back into the cell so that they can be reused. So they get stored back into the vesicles where they were dumped out of. So these are the ways that neurotransmitters are dealt with once they have been sort of excreted into the synapse. Now, amine neurotransmitters contain nitrogen atoms. They often come from compounds that we get in our diets, but they also come from amino acids that we either make or, again, get from our diets. Amino acids are going to be something we haven't looked at yet. Nitrogen containing, definitely, but they are the source of many of these neurotransmitters. And their amino groups are often ionized. So at physiological pH, they're going to have a positive charge on them, and so they'll have ammonium cations. And we'll actually also have carboxylate anions. We'll see that a lot too. Most of our amino acids are charged in both ways. So we're going to take a look at these neurotransmitters here. So acetylcholine first. So acetylcholine is good for communication between the nerve system and muscles. It regulates muscle activity and activation, as well as has memory implications. It's synthesized by an ester bond that forms between choline and acetate. So acetylcholine comes from the acetate ion and this choline molecule here. So this is an ester, not an amide, but it is an amine because it has a nitrogen inside it. Acetylcholine is released into the synapse. It binds to the receptors on the muscle cell and causes muscles to contract. So this is how you activate your muscles with acetylcholine. Now, older adults have not as much acetylcholine. This can have memory effects because, as we know from Alzheimer's disease, um, when you start to have less and less acetylcholine, you start to lose a lot of your brain faculties, motor functions, reasoning. And Alzheimer's disease is just one of many diseases that can affect acetylcholine concentrations. Um, Medications called cholinesterase inhibitors are actually used because acetylcholine is one of those neurotransmitters where in the synapse, enzymes help break it down so that it stops doing its job. And so if you're someone who's suffering from not a lot of acetylcholine or just decreased amount, you don't have enough to really give a good signal in the first place, so you don't want that enzyme rapidly breaking down whatever is there. So this drug is good at inhibiting that enzyme so that it doesn't break down acetylcholine as fast. So acetylcholine, or in this case a cholinesterase inhibitor. Nerve poisons such as sarin, soman, and parathion, these bind to the acetylcholinesterase enzyme and inhibit its action. So this is a different situation. This is if you get injected with one of these poisons, where normally the way that you relax your muscles is that the acetylcholine gets broken down, right, by an enzyme, so then it's not sending the signal, tighten your muscles, flex your muscles. And so that's normally how you stop flexing your muscles, because you have an enzyme that does that. But these poisons knock that enzyme out, so they inhibit its ability to remove acetylcholine from the synapse. So it builds up in the synapse, and it basically stops your nerves from being able to transmit signals. Your body can't relax. You get convulsions, respiratory failure. Um, what's the word that I'm thinking? It's not paralysis. It's sort of like a rigor mortis kind of. Catecholamines. These are the type of neurotransmitter. The most important catecholamine neurotransmitters are dopamine, norepinephrine, and epinephrine. They're related in structure. That's why they're all grouped together. You can get them from the amino acid tyrosine that we get from our diet. Amphetamine and methamphetamine are synthetic central nervous system stimulants. So, and amphetamine and methamphetamine are based on norepinephrine and epinephrine, but they're synthetic, so they've been kind of modified in the lab. They increase excitatory catecholamine neurotransmitters. So that means that they have an effect on these neurotransmitters, the dopamine, norepinephrine, and epinephrine. Let's take a look at those. So amphetamine and methamphetamine will increase the excitatory catecholamine neurotransmitters. Catecholamine just being a category based on their similar structures. Amphetamine is used to treat ADHD, to improve cognition, and to decrease hyperactivity. These are just some structures of these amphetamine and methamphetamine. Now, the catecholamines, these are the dopamine, norepinephrine, and epinephrine. They get made from tyrosine. So tyrosine is this amino acid. It gets converted into something called L-DOPA. So this is the catechol structure. So a phenol, kind of like a diphenol. So that's why they're called catecholamines. Now, L-DOPA gets converted into dopamine in the body, which can then be converted into norepinephrine and epinephrine, or noradrenaline and adrenaline if you use the other common names for those. So that's why they're called catecholamines, because they all are similar like that. And the methamphetamine and amphetamine are excitatory for these guys. So that would cause these to be released more and more frequently. And these are often feel good type um, neurotransmitters. So, dopamine, produced in nerve cells in the midbrain, it's a natural stimulant, energy and enjoyment. This is our reward molecule, basically. It has a role in controlling muscle movement, regulation of our sleep cycle, helps improve cognition, attention, memory, learning. It can be blocked, or the reuptake can be blocked by cocaine and amphetamines, meaning that they are more excitatory. Cocaine causes dopamine to stay in your synapse longer. Amphetamines cause it to stay in the synapse longer. So more dopamine hanging out in the synapse is going to mean that everything feels great. Problem is that when you're not using it, your body has gotten used to that, and so then nothing feels as good anymore because the regular amount of dopamine you get for any random thing is not near as high as what you were getting from your methamphetamine dopamine surge, and so this can be hard to come off of those drugs. High levels of dopamine can actually contribute to addictive behavior and schizophrenia. So L-DOPA. If you have dopamine deficiency, we can actually give you L-DOPA, and L-DOPA is used to increase dopamine levels because it converts into dopamine. Parkinson's disease has people afflicted with Parkinson's don't have the ability to produce dopamine. It could be that they don't have the ability to convert tyrosine into L-DOPA. There could be lots of reasons. So, by giving them L-DOPA, L-DOPA will just convert into dopamine on its own, and they don't have to actually make their own dopamine. Of course, why don't we just give them dopamine, right? Anyways, L-DOPA. What is this showing us? Oh, just as the more vesicles, the more signal, the more neurotransmitter, the better your brain works. Norepinephrine and epinephrine, these are hormonal neurotransmitters. They do have roles in sleep, attention, focus, alertness. Produced by the adrenal glands, usually implicated or involved in the fight or flight response. Epinephrine is synthesized from norepinephrine by the addition of a methyl group. So we saw that they all basically come from dopamine and from L-DOPA. Serotonin, this one helps us relax. This is a sleep chemical, makes us feel calm. Excuse me. This is synthesized by the amino acid tryptophan. So when you eat a lot of tryptophan, or when you eat a lot of food in general that has a lot of tryptophan in it, it can turn into a lot of serotonin. They used to think that tryptophan was really high in turkeys, because after Thanksgiving, right, everybody feels sleepy. Actually, tryptophan is found in lots of things. I think you're probably just sleepy after a big meal, and not so much it being a big meal, turkey specifically. Tryptophan converts into serotonin. Serotonin is implicated or involved in depression. Low levels of serotonin in the brain can often be associated, as it says there, with depression, anxiety, obsessive compulsive disorder, eating disorders, and that kind of stuff. What was I going to say here? So they do make antidepressant drugs like Prozac and Paxil that are called SSRIs, selective serotonin reuptake inhibitors. So they don't just stop reuptake of all neurotransmitters so that they stay in the synapse longer. They selectively only inhibit the reuptake of serotonin so that it stays in your synapse longer for people that are maybe having it broken down too quickly, or reuptake too quickly, or they don't produce enough. And so that's what SSRIs do. They let your serotonin hang out in your synapse a little bit longer. When that is slowed, like I said, it remains longer, and then it's kind of like if you had normal levels of serotonin. Here are the structures of those, Prozac and Paxil. You can see they're both in their amine forms. Histamine. Histamine is synthesized by the hypothalamus from the amino acid histidine. Histidine is the amino acid. This one is an immune sort of neurotransmitter. It's a response to allergies and that kind of stuff. It basically signals your body that you need to start, running your eye, getting itchy eyes, and inflammation and that kind of stuff. It's also released in the cells of the stomach, where it helps to stimulate acid production. Antihistamines, such as Benadryl's anti-Tagamet, these block the histamine receptors so that they don't trigger their intended outcomes. Glutamate, this is the most abundant neurotransmitter. This is used to stimulate most of the synapses. This one is just basically like go, go, go. It tells the next synapse, send your message, send the message. This one has a lot of implications in memory and learning. When glutamate binds to the receptor, it stimulates the synthesis of nitrogen monoxide. It's also a neurotransmitter, so it kind of indirectly or directly releases another neurotransmitter, nitrogen monoxide. So they're kind of linked together. Glutamate levels can be low in certain diseases like Lou Gehrig's disease. I'm sorry, this is an excessive amount, so this is actually too much glutamate. This causes degeneration of nerve cells. Um, Yikes. If the reuptake of glutamate is too rapid, then that's when you have too low of it in the synapse, and that one results in something like schizophrenia. So, glutamate, too much, too little, it's a really fine balance, the amount that you're supposed to have. GABA aminobutyric acid, or, sorry, I said GABA, gamma aminobutyric acid. We know what that means now. Butyric acid is just four carbons like this, right? Gamma means we start numbering from the end, and this is amino, so this would just be an NH2 on the far side of our chain here, I think. Um, Produced from glutamate, most common inhibitory amino neurotransmitter. So this one is sent out when we want the nerve cells, to stop sending other messages. This is like the calm down neurotransmitter. Reduces anxiety, inhibiting the ability of nerve cells to send signals to other nerve cells. Regulation of muscle tone, sleep, and anxiety. You can get GABA from nutrients or supplements. Benzodiazepines and barbiturates, phenobarbital, these can be used to increase GABA levels. So again, these act as sedatives because GABA is involved with relaxing, and so they help increase the GABA amounts. Alcohol, sedatives, tranquilizers, all of these increase the inhibitory effects of GABA. Caffeine actually works against GABA. It decreases the GABA levels in the synapse, and so this can lead to anxiety, or just, what's the word when you can't sleep? What is it? It's not called when you can't sleep. You guys are saying it, but I can't hear you. When you're up all night because you can't sleep, I've got insomnia. That's what it's called. So they can lead to insomnia. Glutamate, yeah, there we go. Glutamate and GABA aminobutyric acid. So I was wrong. It is the salt version, not just the amine version. But, and again, both of these are usually charged in our body. Um, all right, so that's GABA. I think that's it, guys. Actually, so that's basically all the neurotransmitters that we're gonna go over. You don't have to necessarily know everything about them or know their structures or anything, but I hope that going over some of these was at least educational in some sense. Again, we're tying all of these together because they are nitrogen-containing compounds, either amines or amides, but they do have pretty significant roles in our body as both meds and drugs and just regular communication between our cells. So it's good to know something about them. All right. This should be the concept map. Remember the reactions of the amines and the amides. Um, Amines in water, right, as bases, and then hydrolysis of amides in both acids and base. Those are what you should focus on the most, I think. Um, but as always, email me if you have any questions, comments, or concerns, and I will see you guys in our next meeting. All right, guys, take care.